So we stopped on this portion. So we discussed absorptive cells and goblet cells. Yeah, I'll remind you what is the fate of absorptive cells of intestine, small intestine. First of all, it possesses microvilli. You see, one cell possesses microvilli on its surface. Projections of cytoskeleton. We discussed it, but better to repeat. Here inside we have a lot of mitochondria to provide energy to the cell. Here we have smooth endoplasmic reticulum portions, rough endoplasmic reticulum. Remind you difference between them is possession of ribosomes. Yeah, rough is present with ribosomes and functions to produce proteins. And you can and usually it's asked, so why we need to produce proteins here? Because proteins is used to be produced in some I don't know glands, yeah, so which secrete something, but here it's absorbing, yeah, and but actually here we have a very abundant rough endoplasmic reticulum and Golgi apparatus, which notice us that might be the cell produce some proteins, and here is the answer. So actually absorption of lipids, which is happening here, it absorbed in the cytoplasm, and then these lipids should be produced. And here you see these lipids should be associated with our body, because the lipids we absorb, they not always associated properly, with our body. So some compounds can be used in our body, and that's why we need to metabolize them, and we should metabolize them and associate it with our body, so how we need it. And that's why smooth endoplasmic reticulum, mitochondria, which is present here, so they act as reducers and rearrangers of cytoplasmic contents. So, and smooth endoplasmic reticulum. So first of all, you see what happens. These lipid droplets, they are rearranged by mitochondria, might be produced acetyl coenzyme A, and from this acetyl coenzyme A produce triglycerides. And here smooth endoplasmic reticulum can produce cholesterol. So usually triglycerides are produced. So if it is absorbed cholesterol, so then cholesterol should be traveled.
Sir,
And then rough endoplasmic reticulum produces
excuse me.
protein. Yes.
Sir, please share the screen.
Oh, I didn't share it. Okay, my bad. So here, yeah, smooth endoplasmic reticulum here producing might produce not only triglycerides, but also producing cholesterol, and this in a way of droplets. They are present in cytoplasm. Yeah, it's absorbing cells. So I remind you, here we have intestine, hollow intestine. So here from this portion, absorption of absorption of food happens. Absorption of food happens here, and then traveled throughout the cytoplasm. And here, with the help of chylomicrons, so with the help of proteins which are produced on RER, apoproteins, their name is apoproteins, they envelop these lipid droplets and travels it down to the basement membrane. So you see, travels it down to the basement, below the basement membrane, and here we already have lymphatic capillary and blood vessel capillary. And you see, if we have carbohydrates, if we have amino acids, they can easily, they are water soluble compounds, right? And as well, they are water soluble, they are pushed to this lamina propria. Under the basement membrane we have lamina propria, and then from this lamina propria it easily absorbed into the blood vessel, into the capillary, and from this capillary it travels to the vein. So who can remind me which vein is collecting all this blood from the digestive tract? Don't you remember? Okay, portal vein. Now, portal vein system is arised from this portion, so and this will be very important in liver physiology and pathology also. But these chylomicrons, but these fat droplets which are present with apoprotein, here they are very huge molecules. They are pretty large, and when they appeared in lamina propria, they cannot be traveled directly into the bloodstream. That's why they are traveled through the interstitial fluids. And interstitial fluids all over the body, everywhere, is collected into the lymphatic capillaries. So this blind-ended capillaries all over the body, and through these lymphatic capillaries, these large molecules are entering inside. And you see, this protein which envelops this lipid droplet, so is necessary for emulsification of fat. It's like emulsified, and this emulsified fat, emulsified lipid, so is trapped and drained by interstitium and interstitial fluid, and this interstitial fluid is just collected. And you remember what's the color of emulsified fat? Like milk, white. So because milk is exactly emulsified fat, yeah. And this lymph capillary, which is present here, and liquid which is located in this lymphatic capillary, is named as lacteal. Lacteal. So this lacteal system is present in small intestine. So especially, and this will be discussed very soon. And of course, where we can go without goblet cells, without mucus production in small intestine. So small intestine produces a lot of mucus, just in a fate to that might be squeeze out the food. Yeah, so moisturize it. Serves to deepen the traversing of enzymes. So pancreatic enzymes which are produced by pancreas and enter into the duodenum, you know about it. So they have to be enter with the moisturized area, and they're spreading through the liquid, through the mucus, and this mucus should embed every food particulate. And mainly here, of course, goblet cells, they possess a lot of smoothened vascular reticulum, apparatus Golgi, so special apparatus Golgi which produce mucus, and it absorbs fluid from basal lamina, from lamina propria, and secrete it away. And don't worry about the amount of secreted here, so because absorptive cells will return it, because they will return this fluid, this moisture of lamina propria. So intestinal glands. So here we stopped in last time. So the intestinal glands, their name is crypts of Lieberkühn. So that's why don't worry if you hear this strange name, Lieberkühn. So this crypts of Lieberkühn is their just intestinal glands. They consist of absorptive cells, goblet cells, same as we discussed before, then Paneth cells, enteroendocrine cells, and M cells. And a little bit we started here. So this is typical M cell. As I said before, it's work as a lymph nodule. So which traps the composition of microflora of intestine, and here it's analyzed. Here we have antigen presenting cells and lymphocytes which reply on it, and if it's a potential danger bacteria happening here, so we produce antibodies against it, or T cell immunity is activated here. So we react somehow. So that's function of M cell. It's just being a vessel for immunity cells. Here are Paneth cells, as I said before. So Paneth cells, they are responsible. We don't know. It might be secretory granules here. But some other authors they say that these Paneth cells, they collect cell walls of bacteria. And might be both of them right, but anyway, the cells are present with eosinophilic, very highly eosinophilic composition inside. They are present closer to the bottom of the gland, so you see very close to muscularis mucosa. And I remind you that muscularis mucosa is a border between lamina propria and submucosa. Okay, muscularis externa. So wait a bit. We didn't discuss enteroendocrine cells. Enteroendocrine cells, they are similar cells as we discussed in the stomach. So they produce different hormones. One of them is gastrin. We already know it. So same as in the enteroendocrine system of stomach present. And another one is CCK, cholecystokinin. Especially this cholecystokinin is produced in response to fat, in response to any food actually, but especially for fat and for alcohol. So that's why That's why they answer due to presence of something in a small intestine. And cholecystokinin, a main action of them is contraction of gallbladder. Contraction of gallbladder, excessive contraction of gallbladder, so to leak out all the bile which is present there. And I hope you already know what's the bile. Yes, the main function of bile is the emulsification of fat. So, and logically it's right, yeah. But when you eat too excessive amount of fat, this is called cystakinine released, and then gallbladder. If in gallbladder we have a little stones, so they can be abruptly close the gallbladder. So okay, let's discuss it in the respective topic. But enterocrine cells, they produce cholecystokinin. That you have to remember right now. So, and small intestine is pretty similar to along all the lengths of it. Yeah, so you know that the small intestine is the longest structure in our body. It's approximately 3.5 meters. So that's why you understand that it cannot be similar all the lengths, but some very small changes are present there. And here the definition of differences will be discussed. Okay, this is duodenum. This is duodenum, and it was shown in some previous diagrams. So you see, in duodenum, what's the difference between duodenum and the rest of small intestine? Of course, it's possession of Brunner's glands. How to differentiate Brunner's glands from crypts of lubricant? So here you see, this is crypt of lubricant. This is crypt of lubricant. And here we have muscularis mucosa. You remember, muscularis mucosa divides lamina propria from submucosa. Lamina propria from submucosa. And under this muscularis mucosa, we have mucous glands. Pay attention to difference in color of this gland and this gland. They are very highly basophilic because they produce proteins, because they produce some, because their absorptive cells are present, so which also possess roughened plasma reticulum. There, here in lamina propria, we have a lot of lymphocytes because here flora is actively working, yeah, and of course it leads to immunity response to this bacteria. But here you see very lightly colored glands. And the function of these Brunner's glands, we already discussed it a little. In the stomach, you remember, produced a very acidic milieu, and this acidic milieu leaks down into the duodenum. And in the goal to neutralize it, in the goal in neutralization of this Brunner's gland, so produced sodium bicarbonate. So Brunner's glands are main sodium bicarbonate producer in the duodenum. And mucus, of course, mucus. So they are like continuation of pyloric glands of the stomach. Then inner ileum. So I remind you, ileum is the last chamber of small intestine. It is present with Pierce patches. Pierce patches. Might be heard about it. It's lymph nodules. They're lymph nodules. So if here we have diffuse lymphatic system, yeah, diffusely arranged lymphocytes here in duodenum, but here we have these patches. So these patches, it's a collection of lymphocytes. And if we, in case of this diagram, yeah, so here we have predominantly T lymphocytes. Remind you, T lymphocytes which are responsible for cellular immunity. But here, Pierce patches, they are mainly present with the B lymphocytes. And also here we have antigen presenting cells like dendritic cells, and the dendritic cells which are responsible for antigen presenting. So these Pierce patches are typically present in ileum. They are very rarely found in the rest of small intestine. So you see, now we can differentiate duodenum. In duodenum we have Brunner's glands. In ileum we have Pierce patches. If you can see Pierce patches and Brunner's glands in submucosa, you can claim yes, this is jejunum. Yeah. So one more thing, lacteals. Have we discussed about lacteals? I don't remember, but for instance, I will remind you. Lacteals, they're blind-ended lymphatic capillary, which all these chylomicrons, fat droplets enveloped with the protein, which are dropped in lamina propria. From lamina propria, lacteal is arising. Lacteal, so this is lymphatic capillary, which drains all this, especially fat compositions, but also serves to immune function. Because if some injury to this villi happens, so this lacteal is traveled down. Okay, and this lacteal is very well visible on histological slide. So here you see these whitish gaps. Here they are lacteals. They are extremely large in duodenum and jejunum. So that's because a lot of fat is absorbed from these areas. So finally, let's talk about large intestine, colon, histology of large intestine and colon. What first is so you remember the function of large intestine. Unlike small intestine, it's also for absorption, but in large intestine, predominantly absorbed water and salts. Because I remind you, there are a lot of glands which secreted constantly secreted in the small intestine. And of course, we have to return it back to our body. And if it's not returned, so of course it leads to desiccation. Desiccation of the body and water loss. And this happens in some disorders like cholera. Vibrio cholera infection, so which cause the abruption in this absorption from large intestine into the body. Okay, epithelium of large intestine. So you see, same subdivision. We have mucosa, muscularis. So mucosa with muscularis mucosa like a border. Then we have submucosa. Then here lymph nodules are present here. And then muscularis externa and serosa. And somewhere adventitia. So it depends on anatomy. In anatomy, you discussed it. Okay. And very first, what is visible in the large intestine is, of course, glands. They are also named as crypts of Libercium. Also, crypts of Libercium here. And but you see, unlike crypts of Libercium, here we don't have villi. In large intestine, in small intestine, we had villi. So you see, gland is followed with villi. But here we don't have villi, we just have a straight surface of large intestine. It is necessary because water can be absorbed even with this straight layer. But glands are still necessary to squeeze the fecal masses, which might occur here. So that's the idea. In lamina propria, between these glands, below the epithelial lining, between glands, we have lamina propria. Lamina propria is gentle, loose connective tissue, and here we have a lot of lymphocytes. Lymphocytes. For example, if in any other organ we can claim that this amount of lymphocytes is happened due to chronic inflammation or something, but here this amount of lymphocytes is absolutely normal. Because same reason, we have microflora here, and microflora tries to kill us all the time. It tries to digest everything which is on the way, and that's why this immunity cells is trying to save this epithelia, trying to save from any aggressive bacteria which suddenly appeared in a large intestine. Okay. Then muscularis externa. Muscularis externa. So here I wanna explain you one point. Inner circular layer is intact, okay, and outer longitudinal layer is segregated into 3 longitudinal bands, tinea coli. And you discussed the anatomy, You remember the 3 bands which are visible in a large intestine. They are known as tinea coli. Tinea coli, sorry. That are placed equidistantly around the tube. So equidistantly means so with the same distance from each other. And tinea coli, it ties it like in longitudinal manner. And you see, still we have a little amount of inner circular layer, and that's why their binding causes the formation of haustra. So haustra. So this haustra, they like sacs along the large intestine, and that's necessary for vitamin K formation and also in proper formation of physical masses. So either an adventitia or cirrhosis is present depending on the particular portion of the large intestine. So as I said in anatomy, discussed it. Appendix. So histology of appendix. So actually one of the most common histological example so all over the world, because appendectomy, so one of the most commonly proceeded operation all over the world. So appendix. So what's the difference between large intestine and appendix in histological structure? You see, epithelia, mucosa is similar, also same crypts of Libercune with large amount of goblet cells in it. Yeah, I paid you attention. Yeah, so here in these glands, absorptive cells are present predominantly here, on the tip of this, on the epithelium. I mean, on the surface which contacts with the intestinal composition. But here, we predominantly have goblet cells, which secrete mucus. And here you see same picture. Superficially we have so usual epithelia, so which is not very pale, yeah, so not mucosal, but deeping down you see here very pale glands, pale because of mucus collected there. So mucus, goblet cells, you remember, collect mucus on the surface. That's why they look pale in a histological view, because mucus cannot be stained with convenient dyes. Okay. Also here, but here we have a lot of lymphatic nodules, even in lymphatic nodules, and you can confuse them with the same lymph nodules in a small intestine, yeah, and actually they are like similar, but here they are more active. If you remember from histology class, so if you discussed it, if not, so we will discuss it later in cardiovascular system, in a blood lecture system. Lymphatic system is so we have two types of lymph nodules, activated and non-activated. If in small intestine they are usually inactive, so I mean they're straightly composed. So like let me show it here. Yeah. Here appears patches like the lymph nodules. They're straight, yeah. So you see, we don't see the difference between cortex and medulla in Pierce patches in a small intestine. Here we almost always see the differentiation that we have mental zone and cortex zone, and zone inside. This is typical for appendix. Okay, this is histological structure. It's normal histological structure. In case of inflammation, we will see a little different picture. We'll discuss it. Then finally, let's talk about rectum. Rectum is a 12 centimeter long tube, blah blah blah. Mucosa of the rectum is similar to that of the majority of the large intestine. The rectum narrows abruptly to become the anal canal. So here, I wanna pay your attention here. They are known as trabecula. This is distinction between epithelium of colon and epithelium of skin, and it goes pretty abruptly. So here it's visible. Here we have simple columnar epithelium, and here almost abruptly starts the stratified squamous non-keratinized epithelium. Actually, here in some books you can read that slightly transit from the simple columnar to stratified cuboidal. From stratified cuboidal, it changed into the stratified squamous epithelia, and then stratified squamous non-keratinized converts into stratified squamous keratinized, which is resemble skin, as you see here, or even hairs are growing here. Okay, one more thing which you should remember. So this is humeroid plexus. It is humeroid plexus. It's a venous plexus, which is exactly the porta-caval anastomosis. It's a porta-caval anastomosis. So which is it's anastomosis in portal vein system with a vena cava inferior, and through this blood can shunt into the in case of portal hypertension, which we will discuss very soon in lecture. So it shunts there, and it leads to hemorrhagitis sometimes. Okay, this will be a distinct portion of discussion in pathology. Anal canal, so actually also explained. So epithelia changes. You see simple columnar epithelium, then stratified cuboidal epithelium, then stratified squamous epithelium. So simple columnar, stratified cuboidal, then here stratified squamous epithelium. Okay, what means CRG? So I don't know, maybe from other language. Yeah, it's columnar epithelium zone. Okay, I don't know. Okay, here we discussed entire tubular digestive system. So let me open the liver topic. Major digestive glands. So this major digestive glands. So embryologically, they are derived from same endoderm. So actually, I'll send you I think in a Google Classroom or just link to this video where digestive tract is derived from. I have no rights to show this video in YouTube because it's not my intellectual property. But these major digestive glands, which include pancreas and liver, they are derived from endoderm and from digestive tube, and these were just outpoachings. That's why these outpoachings like liver butt and pancreatic butts, there are two butts, there are ventral and dorsal pancreatic butts. They are just small outpoachings, same as Brunner's glands. They are small outpoachings of endoderm, but they are developed into very large organs. That's why we claim them as different organs. But actually, they are derived from kind of same digestive tract. So that's why they are distinct organs. I want to pay your attention. I think in anatomy discussed it, but I want to repeat it again. So that pancreas is derived from two origins. One of this origin, head, and this portion of duct is derived from ventral butt. This ventral butt is attached to common bile duct, and derived from common bile duct actually. And another portion, tail, and this portion of duct, pancreatic duct, is derived from dorsal portion of pancreatic duct, so which is directly bound to the small intestine, to the duodenum. But due to some changes, which you will see in a video I'll share you soon, so this is it happens due to rotation, and mainly is used this duct, and which is common with a common bile duct. So this number 3 is a common bile duct, which transport the bile from liver and gallbladder. And you see it opens with a sphincter of Oddi in a duodenum. And just imagine what happens if gallbladder stuck here. Yeah. So it leads to hypertension in pancreatic duct. Okay, let's discuss it in pathology. So pancreas is mainly exocrine organ. So that's I want to pay your attention. It's mainly exocrine organ. It used to be claimed as a mixed gland, but yeah, it's mixed. It has some small portions of endocrine producing cells, but mainly it is exocrine organ. It producing these different hormones, enzymes, secretory enzymes, and these enzymes are proteins. That's why what we see here, here we have acinar cells, which are typical acinar cells, which were discussed in second semester of histology, or in first semester of histology. These cells are zymogen producing cells, and they possess zymogen granules. They're typical acinar cells with a very thick cells, with a lot of rough endoplasmic reticulum inside it, because enzymes are proteins. Again, I'm talking about it. Enzymes are proteins, and it require very abundant rough endoplasmic reticulum. And this abundant rough endoplasmic reticulum producing enzymes into the duct, and this duct is known as intercalated duct. This is small duct, and then all these intercalated ducts influx into the collecting duct. You see the difference? Intercalated ducts, they're simple squamous epithelium. Interlobular collecting duct is a simple cuboidal epithelium. So, and closer to the end it became, okay, a simple columnar epithelium, a simple cuboidal epithelium is still present. Okay, here we continue, and here are produced enzymes which are necessary for breakdown of proteins, carbohydrates, lipids, nucleic acids. So these main functions. You remember that in a stomach we already had proteolytic enzymes like pepsin, so it was pepsinogen, it's then activated by acid, you heard about it, but still stomach cannot fully destroy entire protein into amino acids, but we need to destroy it till amino acids, or at least oligoproteins, just few proteins in chain should be there. And also carbohydrates, for example starch, or glycogen in the muscles of animals, or might be I don't know, any other starch-like materials should be destroyed by carbohydrates, by enzymes, amylases actually. And you also can remember that in saliva we had amylase, we had amylase, so which also responsible for carbohydrate degradation. But still, this amylase in saliva is not sufficient, and that's why it also needs to be produced by pancreas. And finally, we have lipids. We have lipids in our food, and lipids should be destroyed by lipases, different types of lipases which destroy different types of lipids. But here is the trouble, trouble is that lipids cannot be destroyed in a water milieu. You see, if we have water milieu, aqueous milieu, so fats, they have a tendency to form the globes, so with the how to say it right, droplets, fat droplets. And these fat droplets, they're floating inside this milieu. And how we can get deep into this fat? How can we get deep into this droplet? It's impossible. And enzymes, they work only by surfaces. From chemistry, you remember surface area. How surface area important for a reaction. And this is type for chemical reaction, but it's impossible with a large droplet of fat. And for this reason, we have bile. Of course, you heard about bile. Bile is necessary for emulsification of this fat. So to make this large droplet of fat into the small, very tiny droplets, which increases the surface area of this lipid droplet, and cause the activation of enzymes, and these enzymes will destroy it much easier, and then it is really easily reabsorbed, absorbed by absorptive cells of small intestine. So right today we discussed what is the small intestine, epithelial small intestine. That's why you can understand me now. Duct system, so as said, we have intercalated ducts and interlobular ducts only. So they are the simple cuboidal epithelium, centroacinar cells. Yeah, I want to pay attention to centroacinar cells. They secrete bicarbonates. Their bicarbonates produced, and it is one more gland which produces bicarbonates. Yeah, into the small intestine, which necessary to neutralize the acidity of stomach. You see, we already had pylori gland, which produce bicarbonates. We have Brunner's glands, which produce bicarbonates, and even here, centroacinar cells of pancreas producing bicarbonates. You see how much hardly our digestive tract struggle against the acidity. So three organs, three portions of the digestive tract working against acidity of the stomach. So interlobular ducts lead into one or more excretory ducts that empty into the duodenum. Interlobular ducts, so they enter into excretory ducts and into the duodenum. So histologically, pancreatic gland is very similar to parotid gland. So we didn't discuss what is a parotid gland, but just believe to my words or confirm it by observation of literature. This pancreas and parotid glands, they have simple compound acinar glands. They are mainly compound acinar glands and very basophilic cells with the zymogen granules, which gives this pinkish color inside. Yeah, so that's very brightful cells. But here, what's the difference? Actually, centroacinar cells are present, so which are hardly visible. For example, this is centroacinar cells somewhere, so where I can show it, nowhere else. So might be this under some angle we can claim it's centroacinar cell, but it's not visible. So that's why it's not good thing to distinct from the parotid gland. But secretion of this pancreas is regulated by cholecystokinin and secretin from enteroendocrine cells in the small intestine. We discussed it. Yeah, enteroendocrine cells is a portion of crypt of Lieberkühn, and in the deepness of crypt of Lieberkühn we have enteroendocrine cells which produce different hormones. Here, island of Langerhans, I didn't mention it to you. So you see, here we have very large cells, basophilic cells with very rich cytoplasm, and here we see some small, very teeny tiny cells. And function of these cells is producing very little amount of secretion. For example, pancreas a day producing 1 to 2 liters of juice, but islands of Langerhans produce milligrams of hormones. And of course, for serving this function, it doesn't require large cells. It produces very little amount of protein, named as insulin or glucagon. Okay, let's talk about this. Endocrine portion of pancreas, they are known as islets of Langerhans. You see, there are small clusters of cells richly supplied by fenestrated capillaries. So all features of endocrine system, yeah. They are scattered throughout the exocrine pancreas. So small portions of them are present all over the pancreas. It's less than 1% of pancreas mass. These clusters show no orderly arrangement of secretory cells within the cluster. Okay, which cell types are present in here? They're alpha cells. Alpha cells, they secrete glucagon. They secrete glucagon, and this glucagon main function is destroyment of glycogen. So what is glycogen? I hope you heard. It's a human starch, animal starch, in which carbohydrates are collected. And this glucagon, it elevates glucose level by dissolution of this glycogen. Yeah, we have collections of glycogen all over the body, and glucagon just resorb it and then enter into secrete it into the bloodstream. Then beta cells, they secrete insulin. Beta cells, they secrete insulin. These beta cells, and mainly in clinic, you will discuss about beta cells. You heard about beta cells, and which lowers blood glucose levels, predominant, and this is predominant cell type. You see, glucagon is not the only hormone which increases the bloodstream, increases the glucose level, but insulin is only one hormone which decreases glucose level. And how it works? It works opposite to glucagon. Actually, insulin works absolutely opposite to glucagon. Insulin causes formation of glycogen, especially in the liver and also in the muscles. And we have delta cells. They secrete somatostatin. Its main function is modulate release of other two major hormones like insulin and glucagon. Individual cell types can be distinguished within hematoxylin and eosin. Here we see alpha cells predominantly present around in periphery, beta cells present in a center pool, and D cells, somatostatin cells, they are present so in a border between them. And it also it was not explained here, but there are known as PP cells, pancreatic polypeptide cells, or F cells. So they are the same cells. They also modulate some pancreatic activity here. So they are indistinguishable. So because they have same during the convenience stage, they are invisible. So that's it. That's it about pancreas. Okay, now let's talk about liver. Histology of liver. Liver is located in right upper quadrant of abdominal cavity under the diaphragm. I hope you know it. And functions of liver, they are both exocrine and endocrine function. And please, let's understand it. Exocrine function, you know exocrine function. It's secretion on the surfaces, yeah, secretion on the surfaces, and liver producing only bile. Yeah, it produces on surface only bile. So it works as a very large gland of small intestine, especially duodenum, which produces bile in it. So liver producing, not gallbladder. Bile is produced by liver. So it's exocrine secretion. Then bile acids are producing there, so we'll discuss it a little deeper later. Then we have endocrine function, and actually liver can be claimed as the largest endocrine organ of the body, and main producing endocrine organ of the body. So why? Because endocrine function of liver including not only a hormone secretion, but also secrete plasma proteins. And if you're confident in a structure of plasma, so you remember that almost 10% of plasma is composed of proteins. They're like albumin, globulin, and fibrinogen, and which one of the clotting factors. Also, it produces lipoproteins, which rearrange these chylomicrons. You remember chylomicrons, which were absorbed by absorbing cells of small intestine. These chylomicrons are very dangerous for blood vessels, and that's why liver rearrange it in lipoproteins. Okay. And then additional functions include the metabolization of digested food, storage of glucose as glycogen, and detoxification. So these functions you should remember very clearly, because in pathology of liver, so you have to mention that we have so we will be troubles with exocrine function, with endocrine function, with the metabolization of digested food, storage of glucose as glycogen, and detoxification. All of this included in liver pathology. So here you see, yeah, interior vena cava. So this is structure, general structure. We have diaphragmatic surface, it's very straight, yeah. Visceral surface, which possess some incisions because small intestine is projecting it. So ligaments, so in anatomy you discuss it. But inside histologically, you remember it possesses lobules with the portal triads in it. So here we have to discuss it. Okay, so first of all, let's talk about blood supplement of liver. Blood supplement of liver goes from two main sources. First of all, is from portal vein, and second is from hepatic artery. Portal vein, hepatic arteries, so pretty logically understandable. Yeah, we have an organ, we have to supply it with arterial blood and to supply it with oxygen and then return it back in the way of vein. So from the liver, so drains the common hepatic vein, and this hepatic vein influx. So it's very short vein, so it influx directly into the vena cava inferior. Okay, and but here also influx vein. So it's very strange, yes, that vein enter inside the organ. You discussed in physiology, I understand, but for some who is not confident, and explain it. Portal vein enter into the cell, enter into the organ, and here it spreads into capillaries. And these capillaries are known as small venules, first of all, inlet venule, and then into the sinusoids, perpendicularly to this venule. You see, when sinusoids, they're sinusoid capillaries actually. It's sinusoid capillary, so which travel through the central vein. Okay, so that's how blood supplyment of liver goes, and we can distinct entire liver into these lobules. So that's why in the final destiny, so enter into the tissue going from portal vein and from hepatic artery. And this vein and artery, they're present very nearby, and they fuse here their products, so their blood. And here we have mixed blood, so from vein and from artery. And artery, so it's understandable the reason of it, yeah. So just embryologically, arteries come inside the portal, inside the liver, because before no portal vein were exist. And also it requires oxygen, because portal vein it still contains oxygen, because it collects blood from entire peritoneal cavity, from all organs of peritoneal cavity, like small intestine, large intestine, stomach, and even it also contains not peritoneal organs like spleen and pancreas. It collects this blood from these organs too, and all of this blood influx into portal vein, and from portal vein it travels through the liver interstitium. So, and all these functions became clear, yeah. All these functions became clear, like metabolization of digested, food. So because all the food which is absorbed from portal vein enter into the liver. Storage of glucose and glycogen, when you eat something sweet, it contains a lot of glucose, it absorbed in a small intestine, but it shouldn't increase our glucose level, yeah, because glucose level is very necessary to be maintained on the same level, throughout our life. And that's why storage of glucose as glycogen is a very important function of liver. And detoxification, so because we eat a lot of waste products. We eat a lot of junky food, or even in healthy food, which is claimed as a healthy food. So also contained a lot of toxins, and liver detoxify it throughout this short portion of sinusoids. That's function. So actually, that's the main function of liver. So that's it's to rearrange all these contents which are coming from portal vein, a little bit from hepatic artery, because in the rest of our body also waste products are produced and can be entered with the hepatic artery too. So, and this short prolongation of sinusoids is the main goal of this liver. Like alveoli is the main goal of lungs. So this main goal of liver is this territory. Okay. But it's two vessels here, yeah, portal, but it's named as portal triad. But actually nowadays it's more commonly named as portal tetrad. So, but let's talk about classical classification. It's a triad because here we have bile ductal. So about this bile ductal, just keep in mind, but I'll explain it in details a little later. Here is the structure. Again, you see, it is not so strictly as it's shown here. So you see, it's Here it looks like glands. Yeah, it looks like glands. These cells are hepatocytes. Hepatocytes, these pinkish cells, they are hepatocytes. Between them we have sinusoids, and into the sinusoid influx artery, portal vein, and they are intermixing here. And final destination is the terminal hepatic venule. So you see, it's not so strictly shown as it's shown on this diagram. Yeah, so yeah, perpendicular here like this. No, actually it's going this way. Yeah, so we like in nature. Yeah, so we are natural product. We are came from evolution. Yeah, derived from evolution, and of course we have same natural pictures inside our body. And here it's not straight, but it's coming in this direction from portal vein, from hepatic artery, very way coming into the central vein, slowly coming to the central vein, and which allows these hepatocytes to act its function with make the function with these sinusoids, with the blood which present in the sinusoids. And here you can mention another thing, bile duct. You see, bile duct is arising also between hepatocytes, but from inner portion hepatocytes. So here we have what, sinusoid, and between two hepatocytes starting the bile duct. So actually, so might be you heard about this position, so here I have to mention it. So you heard about this point that liver is a very rapidly regenerative organ. Yeah, liver can reproduce itself from small portion. Even 1/10 of this liver can produce a rest of liver. Yeah, so that's why donation of liver is not so dangerous. Yeah, they claim. Actually, kinda yes. Yes, it's kinda true, but with a few. If you're healthy, yeah, it's kind of true. So you can donate your liver, so and it will be reproduced, but not reproduced in the same way like a tail of reptilians, but it functionally increases in size and can cover function of rest of insist portion of organ. But here you see, here we have cells of bile duct. Cells of bile duct. Here, hepatocytes. But you see, their origination is similar. And actually, here we have cells which are cells of bile duct. They are known as cholangiocytes. Cholangiocytes. Here, hepatocytes. Cholangio is chole. It's like bile, angio, vessel, sites, cells. Yeah, cholangiocytes and hepatocytes. And cells which are present between them, so which are non-differentiated, they are like stem cells for both, for hepatocytes and for cholangiocytes, cells of bile duct. And the point is, you see, these cells are mitotically active. These cells can produce other cells, can produce hepatocytes, but here cells, they can't produce hepatocytes, they can't divide anymore, and you see this direction. So, so here we have very rich supply node. So because all the oxygen is located near this area, all the nutrition comes in this area, and here we have reproduction of cells. Okay, rapid growth going here, but cells which are present here, they suffer with hypoxia because most of oxygen is already distributed in first portions. Nutrients also can be absorbed before coming here, and you see these cells suffering with hypoxia and insufficiency of food, and what they do, they die. And that's why and this direction you see, so from this portion cells gradually come to this portion, from the triad portion it comes to central vein portion, and that's how refreshment of liver goes. And if it's required, we have liver growth factors, and so hepatocyte growth factor, so which and epithelial growth factors, which cause the so in case of insufficiency of liver function, So they start to produce more hepatocytes than necessary directly to this portion of liver. So that's how mechanism of rehabilitation of liver goes. Okay, blood supply and drainage of liver. So discussed functional microanatomy. Yeah, discussed. Here I want to show you this diagram. So you can observe this lecture you can just play stop during the observation on YouTube. I want to explain you this structure. You remember sinusoid, yeah, from this diagram. So this is sinusoid in a macro view, large view. This is hepatocyte, yeah. So please, I want to fix this image for yourself. And then here, this is same sinusoid, but under the closer view. As well as sinusoid. Sinusoid means discontinuous capillary. As well as discontinuous capillary, it contains same endothelial cells, but with very large gaps between endothelial cells. They don't have very tight connection between them. And lamina propria, basal lamina is not continuous here. So you see, between endothelial cells we have large gaps. And which portion of blood enters here first? Of course, plasma, and sometimes even cells can penetrate this space, can penetrate under this endothelium. And that's exactly what we need. Because under these sinusoids we have special space. This space is known as space of DC. Space of DC is a loose connective tissue. It has very common features with lamina propria, and its function is absorption of this plasma, absorption of plasma components. Because I remind you, from portal vein, usually, especially through the plasma, come in a lot of waste products, come in a lot of nutrients, come in a lot of proteins, and all of them are passing here, and they can penetrate this space of DC. And from this space of DC, so these nutrients are absorbed by hepatocytes, and these hepatocytes even have microvilli on the surface to absorb more products here. And then glucose is absorbed here, fats are absorbed here, amino acids are absorbed here. They collecting into the cytoplasm of this hepatocyte and provide its function. Even toxins also absorbed into the space of DC and also absorbed here. But you can say, what about cells? But why cells are not penetrating here? Actually, cells are also observed, but not by this cell. And this is like very not properly working filter. It filters only plasma, but cells are very rarely enter inside. But cells are also digested by liver. And here it is. It is macrophage. It is known as Kupffer cells. It's a Kupffer cell. This Kupffer cell is a macrophage which can trap senescent erythrocytes and can serve as a spleen. Same as spleen, it also traps the senescent erythrocytes, and the senescent erythrocytes are destroyed until amino acids and bilirubin and also into the iron and travel back into the bloodstream. So its functions of spleen we will discuss more detailized in blood topic. But still, you remember that graveyard of RBC is the spleen, but 10% of this function is on these Kupffer cells. But also another function of these Kupffer cells, everything which is large enough not to pass into space of DC but need to be deteriorated. So we can say that these Kupffer cells destroy it into the small portions, rub it into small portions, and then turn it into the space of DC, and then hepatocytes provide a rest of function. Okay, so as I said, lipids absorbed here, chylomicrons absorbed here, then with the help of mitochondria, smooth endoplasmic reticulum, and with rough endoplasmic reticulum is produced another proteins which produce 4 types of lipoproteins. actually. They produce necessary amount of lipids for us and proteins which carry them. So they are low density lipoproteins, very low density lipoproteins, high density lipoproteins, and transient density lipoproteins. It's not exactly a right way I say it about it, but in biochemistry, in details, you should have discussed. You will discuss it later in clinical biochemistry. Yeah, and here it is. And here these lipids are produced and then returned back by the same way. So these lipoproteins they enter into this area and then return into the bloodstream. You see, that's one of liver function. Secondary, toxicants. What happens to toxicants? So okay, first of all, with amino acids. Okay, from the extract, amino acids enter in here in capillaries into sinusoids. Amino acids enter inside. Here we have rough endoplasmic reticulum. Here we have genetic material which is responsible for formation of globulins, albumins, and fibrinogen. And proteins are produced and returned back into the bloodstream. Pretty easy, understandable. Fats we discussed. Cholesterol. Cholesterol which enters here with the chylomicrons. Some of them return back in lipoproteins, as I said. But also they can be drained into this bile canaliculi. And this bile canaliculi is separated from space of Disse with the help of zonal occludens, which are like, so in easy words we can say it's desmosomes, which are surrounded and don't allow bile canaliculi to enter into the space of Disse. But spoiler, in pathology we'll discuss when this bile canaliculi hypertangent and this zonal occludens ruptures and this bile leaks into the space of Disse and into the blood, and which leads to, someone know, jaundice. Leads to jaundice. So when skin become yellow. Okay, and then what happens to carbohydrates? Carbohydrates, when they enter here under the control of insulin, without insulin it doesn't work, but with control of insulin, these carbohydrates forming the glycogen granules. These glycogen granules, which are collected here, they are collecting carbohydrates inside the cells. And under the work of glucagon or adrenaline or epinephrine, yeah, activation of these hepatocytes happen, and glycogen destroys still glucose and return it back. So you see, it's coordination center. So it's a center by which we regulate the amount of glucose in our bloodstream. If it's too much glucose, so islets of Langerhans, beta cells of islets of Langerhans producing insulin, and blood directly from the pancreas enter into the liver, and from the liver it is and in the liver this insulin which is produced by pancreas, so provided its function, collection of glycogen. Okay, finally we discussed about amino acids, proteins, and carbohydrates. So here we have a lot of mitochondria, which is necessary because it's actually hepatocytes are factories of our body. It produces a lot of materials, yeah, and the functions are very distinct, but very interesting, and in the same way. And here, this cell, we just avoid attention of this cell, Ito cell. Ito cell is a mesenchymal cell. So this mesenchymal cell, which is same as fibroblasts, which are mainly producing fibers. But here you see is becoming fat fibroblast. I mean, these fibroblasts collect fat droplets, especially with vitamin A storages. So because vitamin A is a lipid soluble compound, it stores fat with vitamin A inside it, and if it's necessary, retinol is produced and returned into bloodstream. So first of all, passing through the hepatocytes, and then it enters into the bloodstream. But okay, not interesting. But much more interesting for us that these Ito cells are very sensitive for inflammation, very sensitive for different stimulus. These Ito cells can be rearranged back into mesenchymal fibroblast, and this fibroblast producing collagen, yeah, and they produce collagen into space of Disse space. and this collagen makes it thicker, and it causes fibrosis, yeah, and this fibrosis in liver is named as cirrhosis, yeah, cirrhosis of liver. So that's why eto cell is might be responsible for cirrhosis formation. Okay, 1 second. These eto cells, yeah, as I said, they're fibroblasts, and especially under the influence of inflammatory reactions, like Kupffer cells are macrophages. They provide the same reactions, like you remember macrophages from previous semester. Remember macrophages, they have two mechanisms of activation: classical activation and alternative activation. Classic activation of macrophages happens in acute inflammation, and alternative activation goes in chronic inflammation. And then when it's chronically, so in chronic inflammation, alternatively activated Kupffer cells, they produce fibroblast growth factors, platelet-derived growth factors, and also transforming growth factors. And it leads to reorganization of eto cell into fibroblast. So it's genetical changes, of course, happening here. They have triggers for to change into fibroblast, and it leads to, of course, very commonly it leads to cirrhosis, very common disease. So this is a real picture. So this canal of herring, so it's herring canal, it's a bile duct. Intrahepatic bile ductal, periportal space of mole, so that's, yeah. Flow of bile from liver. So how bile comes from liver to the to outside? First of all, these bile ductals, they are collected into bile. So let's turn back. So here, bile canaliculi comes into bile ductals. So here it's written like bile duct, but it's bile ductal is small. But then these bile ductals are collected into bile ducts, and we have common. So it's a right bile duct, left bile duct, then common hepatic duct, and then outpouching of this common hepatic duct, forming the gallbladder, and this common hepatic duct joined with the cystic duct forms a common bile duct. This common bile duct, facing the sphincter of common bile duct, or named the sphincter of Boyden, and then finally it enters into the sphincter of OD, hepatopancreatic ampulla sphincter, and then it with a major duodenal papilla, so anatomy discussed, enter into the duodenum. You see, this is a normal pathway of bile excretion. I remind you that bile is not only responsible for excretion of bile salts, but also responsible for excretion of bilirubin. Bilirubin is in pathophysiology we'll discuss. You should answer to me, because in physiology you discussed it before. Then another function. Yeah, I forgot to say one more function. Yes, sorry. Toxins. What happens in liver with toxins? When toxins enter into the cell, it's absorbed, and here we have smooth endoplasmic reticulum, and inside the smooth endoplasmic reticulum we have cytochrome P450. Cytochrome P450 is a cytochrome which is responsible for attachment to lipid-soluble products, some water-soluble components, like glucuronic acid in case of bilirubin, sulfic acid derivatives. So to rearrange, so the main idea of this cytochrome P450 is to rearrange lipid-soluble molecules into water-soluble molecules, especially toxins. And these lipid-soluble molecules, because lipid-soluble molecules, they are very dangerous in the tissues because they can pass through the cell membranes. Yeah, because cell membranes are very well protected from water-soluble material like because they have a bipolar origin. They have a positive or negative charge, or both of them in some manner. So that's why we can avoid ourselves from it. And most products of the earth, they have charge. Yes, that's why they're water soluble. But in case of lipid soluble small compounds, they can penetrate through the cell membrane and can destroy innate structure of cytoplasm. So that's why main goal of liver in this way, in antitoxicant function, so it's to rearrange this lipid soluble material into water soluble material and return it back to the blood. And what happens to this back to the blood toxicants? Of course, they are excreted by kidneys. So that's why liver work together with the kidney. Liver can't excrete all material. They produce only bile, and bilirubin is produced outside directly by liver. But main other compounds, main other waste products which are produced by liver, detoxified waste products which are produced, they return to the blood, and from the blood later after it they excreted by kidney. Let's talk about gallbladder. Take a look, structure of gallbladder. Resembles something, yeah? It resembles, of course, it resembles small intestine, but it doesn't contain crypts of lubricant. It doesn't contain lacteals. So we have some villi. So it's like poor small intestine. So it requires not so excessive absorption. It requires not so excessive moisturizing. So that's why goblet cells amount are very decreased. So we have no lacteals because fats are not absorbed here. So, and here we have very thick muscular layer, and this muscular layer is not very well arranged. So it's arranged very well, but it looks like irregular network surrounds the gallbladder. You see. So let's step by step. Mucosa, simple columnar epithelium with short macrovilli. We understand it. Lamina propria, same as everywhere. Muscularis mucosa is not present. It's not necessary because here we don't have to arrange any villi. So it's thrown into complex irregular folds that are particularly evident when the gallbladder is empty. Some mucosa, smooth muscles arranged in irregular network, and serosa. And you see, serosa is covering the portion which is out of liver, but portion which is close to liver, which attached to liver, it is covered with adventitia. That's a histology of gallbladder. So here we have glands, of course, a little bit of glands, and very thick muscularis externa. That's main portion, but it's not compulsory. Here we have without mucosa, mucosa without glands. So that way you see it's like poor small intestine structure, simple columnar epithelium. That's it for today. Thank you for attention. If you have any questions, you can ask me now. So under the record, if you don't want, so I'll stop the record and you can ask me your question. Any question? Thank you for attention. See you next time.